So this is the landing page to the reference database pipeline. gttn.treegenesdb.org is the link to get to this page. So from here, as long as you're logged in and you have an account, you can submit your reference data. Once you get to this page, you'll be able to either create a new GTTN tips submission, or you can pick up where you left off an old submission if you have those. So the first page here covers a lot of basic information about your submission. So that includes the submission name, the date that the analysis took place, DOIs that are related to your data, the original database URL if the data came from a different database originally, stuff like the project name. So like this would be either a funding agency or grant number. What's important from this section is that you select a submission type. So this indicates the type of trees that you're submitting, whether they're new or existing already in the database or a mix of those. The next section is data permissions. So this dictates who's going to be able to see your data once it's been submitted and approved. So there are a few options here that are pretty flexible. First is public, so this will be visible to everybody who goes to the GTTN website. All registered GTTN organizations. This of course shows the data with shares the data with every other organization that is part of the GTTN network. Law enforcement members only. Selecting this option would make your data only visible to users who have the law enforcement role. Select organizations. So this is useful if you'd like to share your data with multiple organizations within the network, but not necessarily all of them. So from here, you can click as many organizations as you like, or you can just share it with the organization that your account is actually paired with. So for today, I'm just going to share my data with this organization. And then there's finally a disclaimer here. It's a placeholder for right now, but it basically says that you're consenting to submit your data and have it be shared with the people that you said you're going to be sharing with. So the second page is the species information. Here you can add as many or as few species as you'd like. The species is autocomplete. So if I type in E, it autocompletes to any species that start with E. Today I'm going to do Absolia africana. And then the data types section where you select the different types of whether you're giving us sample data or the different types of reference data that you'll be uploading today. Okay. So this is the tree accession and sample information page. This has two major sections. The first one is the tree accession information. This is mostly just one file upload, but this is a dynamic file upload. So what that means is after you upload your file, you'll see a preview of it. This is the first few rows of my Excel spreadsheet. And in the columns of each of the preview, there's a drop down, and this helps us decide what data is in each column of your file. This allows us to take in a lot more different formats of files, and we don't have to rely on regular expressions to try and guess what the types of data are in each column of your file. So this file upload requires a tree identifier column and some location columns. So we have the options for tree identifier. You can specify the location of each tree either with GPS coordinates like I have here. We also offer options for specifying location via the country and state, county, district. You can specify a population group, and then you'll get a little section down here that asks for the location of each population group if your trees are grouped by location. We also accept forest IDs and barcodes, and then there's an optional column for species identification confidence score. So once you've selected columns for your tree identifier and the locations of your trees, you can click this button, and it allows you to see your trees on an interactive map. So it's fully interactive. You can scroll around and zoom in and out, and you can also click on individual data points to see the name of the tree that we think is at this location, as well as the location that we think it's at. So this is super useful for a quick check to make sure that none of your trees end up in like the ocean, for instance. That generally would mean that there was a problem either with your file or we parsed it incorrectly. So this is just a helpful way to a little sanity check. So next is the sample information section. This is pretty similar to the tree accession information section, as it is mostly just a dynamic file upload. So similar to the tree accession portion, after you upload your file, you'll get a preview, and then we just need to know the types of data in each column. So for this file upload, we need to have a sample ID. We need to know what the tissue type is of that sample. We need to know the remaining dimensions or volume of that sample. We need to know who collected the sample, what the source of the sample was. So this can either be a tree ID that matches a tree ID in your file that you submitted up here, or it can be the ID of another sample. So if you had like a sample coming from a sample, then you would just have to specify the ID of the sample that one came from, the dimensions of the sample, and then finally the sampling method. And then after you've indicated all of that, we just need a little bit more information about your samples, like when the samples were collected, whether or not they've been analyzed, storage location. So this can either be, there will be a dropdown of registered GTTN organizations, or you can specify other and put a different storage location, and then the technician name and email of the person who collected the samples. And then finally down here, this little box indicates whether or not these samples can be shared with other people within the GTTN network. And this will come in handy later when we talk about data certifications. Next. So this is the final page of the pipeline that actually has you inputting data, and it's the largest page. So this is where you'll enter your actual raw analysis data. So that includes your DART data, isotope data, anatomy data, or genetic data. So at the top is a little checkbox that indicates whether or not the reference data for the submission should use the same permissions as the rest of the submission. So if I were to uncheck this, it would have an option to either select different organizations that can see this data or restrict it to only the current organization. So if I was to switch to only the current organization, then the metadata for this study, so the information about the species, the type of data, the location of the trees, the name of the submission, all that would still be visible, but the actual raw data would only be visible to the current organization. But for now, I want to keep the same visibility permissions. You can also select to not provide the reference data for this submission. So if you wanted to indicate that the submission had DART data, but you don't want to upload it to the GTTN reference database, then you can click this box here and skip DART data. And this option exists for all of the different data types. So isotope, genetic, and anatomy also have a box like this at the top of each section. And it all works the same way. Okay. So first I'm going to cover the DART reference data. There are two files that need to be uploaded here. So the first is the DART metadata spreadsheet. It's a dynamic file upload, as you can see, the preview and the different column data types. For this file, we need to know the name of the analysis lab, the spectra ID, the spectra gatherer, the type of DART TOFMS, the parameter settings for that DART run, the calibration type for that DART run, and the sample ID that DART run is associated with. Then we need an archive of the raw DART data. So this is the archive that I have uploaded here. It should be a single folder that contains a bunch of text files. Each text file should be named the sample ID.txt. So for example, this one, TW58, matches the sample ID up here for that DART run. And then in each text file, there will be a couple lines of calibration information followed by the actual raw DART data. So if you format this archive correctly, then when you upload it, the pipeline system will extract it and be able to parse through all of this data and then load it into the database properly. Next is isotope reference data. First, we need to know whether or not an increment core was used for sampling. And if it was, we need to know the length of that increment core. Then we need to know all the different isotopes that were used. So today I'm using 13C and 18O. For each isotope that you used, we need to know the standard and the isotope type, whether it's whole wood or cellulose. And then finally, we need to know we need to have the actual isotope raw data. So for this file, we offer two different formats that you can upload your spreadsheet. The first contains a unique sample ID followed by all of the isotope data for that sample ID, separated out by columns. The second, the sample ID may appear more than once, and there's only isotope data for one isotope in each row. The reason we offer both of these formats is we found that lots of people had files of the type 1 format and lots of people had files of the type 2 format, and so we figured we would just try to accommodate both of those. So for today I'm using type 1, so I just need to indicate the column with my sample ID and the columns for my two different isotope types that I am submitting today, and then I'm done with isotope. Next is the genetic reference data information. Today I'm just using SSRs. If I select SNPs or other genetic markers, I'll get a bunch of different questions, but for today I'm just going to use SSRs. For SSRs, we need to know the sequencing, the name of the SSR sequencing instrument, ploidy of the species that this SSR run was done on, and then the actual genotype assay file. Then optional fields include the description of the SSR marker, the DNA quality, and the DNA extraction method. Finally is the anatomy section. First, this section is the anatomical characteristics. This is a bunch of metadata about the species that you've indicated you're uploading anatomy data for. These sections were all provided to us by an anatomy expert within the GTTN network. But these are all optional, so I'll skip them for now. The part that's not optional is the microscope slides. So this is where you'll actually upload your microscope slide pictures. So you can upload the file. We require a short description of the slide image that you're uploading, and then you can optionally add a photo credit. So once you've got all that data entered, you can click Review Information and Submit. It will bring you to a summary page. This page is just similar to the thumbnail map on the tree accession page. This is just a sanity check to look over and make sure that all your data looks correct. The submission name, the reason for the submission, the number of trees, and we intend to add a little bit more information like the number of DART runs or number of genetic markers or isotopes that we found, just to make sure that all the numbers look right. Once you've verified that all of this information is correct, you can click Submit, and then you'll just need to wait for approval from the primary contact of your organization.